So I'd like to welcome everybody to the annual Arthur Ludwig Lectureship in Humanism Medicine. And I'd like to invite Emily to introduce our speaker this morning.
Thank you, Dr. Rauscher, and good morning, everyone. It is a pleasure to introduce the annual Arthur W. Ludwig Lectureship in Humanism and Medicine this morning. This lectureship is part of our Advancing Idealism in Medicine initiative, which was founded by our residents and includes a grand round speaker series inviting inspirational role models in the practice of medicine. Dr. Ludwig arrived at Mount Sinai as an intern in 1943. After serving in the Navy in World War II, he completed his training in internal medicine, followed by fellowships in both pathology and endocrinology. He remained affiliated with Mount Sinai throughout his entire life. Dr. Ludwig served as an attending in the endocrinology clinic and as chief of the outpatient department diagnostic clinic. He was regarded as an outstanding diagnostician and a strong patient advocate, the provider of superb and compassionate care, which earned him the highest level of trust from his patients and their families. Today, I'm happy to present Dr. Charlotte Cunningham-Rundles with the Arthur W. Ludwig Lectureship Award. And I have this plaque here. Dr. Cunningham-Rundles is the David S. Scotsman Professor of Medicine and Professor of Pediatrics. She established and directs the Immune Deficiency Program at Mount Sinai, is a past president of the Clinical Immunology Society, and has served on the board of directors of the American Academy of Asthma, Allergy, and Immunology. She has also served as the chair of various committees for the NIH, the FDA, and the National Aeronautics and Space Administration. Dr. Cunningham-Rundles' accolades and scholarly and professional accomplishments are truly too numerous to list in a grand rounds introduction. So instead, I will take a moment to just share a brief anecdote from when I was a medical student and I shadowed Dr. Cunningham-Rundles in her clinic. In a single afternoon, no fewer than 4 of her patients had come from outside of New York to come and see her specifically, some flying in just for this visit. Most were suffering from extremely rare illnesses, and others were still searching for a diagnosis. Between each patient, she told me a bit about their backstory and what big life events they'd recently celebrated, and what research was being done to better understand their condition. And from the person at the front desk to the immunology fellows to every parent I spoke to, they all said the same thing. "Oh, you're working with Dr. Cunningham-Rundles today. You're so lucky. She's the best there is." And it's my great pleasure to introduce today a person who has done so much for so many, for patients and trainees, and really the medical field as a whole. Thank you for joining us, Dr. Cunningham-Rundles.
Thank you, Emily. That's over the top, but thank you very much for that very warm introduction. These are my disclosures, and I'm not talking about any products or any medications today, so these things are really not pertinent at this point. This is a very nice picture of Dr. Ludwig. I'm sorry it's black and white. He's here in the center having very informal rounds with a number of his colleagues. I thought it was a very nice shot of exactly the type of physician that Dr. Ludwig, I think, was. As Emily said, I put a small little bit of bio on this slide here, but I think Emily went through this. One of the quotes from people working with him in the last portion here said that Dr. Ludwig was really a very notable practitioner in the art of medicine. He listened to the patients and he was a superb physician, a marvelous diagnostician. His judgment was uncanny and he insisted on the very best for his patients. I thought that was a wonderful quote. The other thing which I looked up was exactly what did Dr. Ludwig find as interesting himself. And this is one of those things that I think that all of us would very much have enjoyed talking to him, because he had a great interest in how estrogens and testosterone worked in animal models. And that was very pertinent because he found that that was why the rooster's comb grew due to androgens, for example, and estrogens, on the other hand, would take that back and make that to regress. He was also very, very interested in the deposition of extracellular matrix fluid, especially in exophthalmos and also in myxedema. And I show you just some pictures of what he was looking at, but he was the one who figured out and had to do with the finding that it was hyaluronic acid in both situations. It's just one of those things in medicine where unusual things come together, but then they really illuminate what's going on in a much bigger context. And I find that very interesting. So today, what I was thinking about talking about, this is a lecture in humanism, but one of the things that we've all been thinking about so much is all about antibody memory. We've all been immunized so very, very much in these last few years. And the question is, well, what good did it do? And how long is it going to last? And you have to go back, and this is a slide I got from the CDC, which just says such an astronomical number of vaccine doses have been administered in the United States. And I know, and everyone here in this audience has had 2 or 3 or even perhaps 4 doses of vaccination in the last 2 years. So it's really on our mind a lot. How long does this antibody going to stick around, and how good is it? And what's it going to do for us? So what is it supposed to do? Well, what it is supposed to do, of course, is give us IgG antibody binding to the spike protein in our blood. Again, a very nice slide comes from the University of Pennsylvania Vaccine Center. And the ones that are in red, those red dots, are people who already had COVID themselves and got the vaccination. And the blue ones are those who just simply got vaccinated. But it's interesting, everyone is ending up having a great deal of antibody over the course of, you can see, 1 month. So in 1 month, a lot of antibody has already been started to have been produced. And by the way, this is a log scale, so that's really quite a bit of antibody. Now, what's interesting also is that if you look in the blood, what you can find is that antibody-making cells in the blood, B cells, are able to bind to the spike protein itself. So these are called memory B cells because they have gotten their job assignment straightened out. They know what they're supposed to do, and that's the only thing they're ever going to do. Remember, B cells have only one job in life, and that's to understand their antigen and to go after it. And after that happens, they don't go back. They can't change. They've already made up their mind. They've got their diploma. And so what these are diploma-bearing B cells that say, I know what spike protein is. And again, here we are at 30 to 40 days or so after the vaccination. But what's the question? The question, of course, is, okay, that's great. How long is it going to last? And then all these questions, I took these from various websites and from newspapers. And I think everybody here is probably getting exactly the same questions in their own practice. In other words, how long is antibodies going to last? How long will it happen after I've been vaccinated? How long does the immunity last? Will it last a lifetime? Very, very ambitious statement over here. COVID immunity, can you catch it twice? Well, actually, we know you can. Do I need a 4th vaccination? And so forth. And then the experts weigh in. I'm not going to tell you that I'm an expert on this topic, but I will go into what we know and what we can gather. So the question is, how long does antibody last? This is kind of an astonishing event here. Thucydides in 430 BCE noted very carefully and wrote this down that there was a infection that killed about 1/3 of everyone in Athens at the time, astronomically serious infection, clearly, and the same man was never attacked twice, at least never fatally. So even Thucydides felt that antibody memory was a possibility. Once you had it, you weren't going to be getting it again, at least not in a very serious form. Now, he wrote that down. He didn't know what the infection was at the time, but in this graveyard in Athens, those bones have been exhumed, and we know that the typhoid bacterium has been found. And so this was actually typhoid, and we actually now know that. It gave us an interesting point of view that, 2,000 years ago, there actually was a point of view about this, and I find that really pretty amazing. So what I was going to talk about and think about today is history of vaccinations. And you have to understand that I really think antibody making is the most amazing thing a human being is doing. Being an immunologist, you have to expect that I would probably have a viewpoint like that. But B cells really go through an amazing routine of doing what they do. Secrets of plasma cells, and then the questions about antibody vaccines. And then, of course, in the laboratory and in the clinic, what I spend a great deal of my clinical time on, okay, well, that's wonderful. Normals do that, normal people, but what goes wrong? what can go wrong? And then, of course, ending up with things about what genetics has now told us about these antibody secrets. Oh, let's see, sorry. It's one of those glitchy things that you don't get to be. Let me get back to my slide. Here we go. So what's going wrong? And tell us something about how these antibody secrets work. This is this always amuses me. This is actually the first vaccine laboratory on Earth. And this is a hut in the garden of Dr. Edward Jenner. And it happens to be in the western part of England near Berkeley Castle. I visited there. So if anyone goes to England, they should probably go and visit this interesting little hut. And the reason why it's a hut in the garden is because Mrs. Jenner said, "You are not doing that in my kitchen." And so he had to build this little laboratory, so to speak, in the garden. And it still is something that you can visit. It's now a museum. And the first vaccinations took place in 1798. I think everyone knows that this was based upon the relationship to cowpox. And of course, when taking cowpox and putting the variolation, which would be the scabs and the material that came out of the pustules of people who'd suffered from cowpox, and immunizing, it was pretty unpopular, as you can see. And people, of course, were terribly scared. I think we in a way, we find this similar to the rumors and the discussions which take place with COVID vaccination, don't we? But as you can see, this was not a new idea. The wonderful effects of the new inoculation would be perhaps to turn people into somewhat of a monster situation. So again, very old print, but it goes to show you vaccine difficulties were common at that point as well. But of course, it turns out to be enormously beneficial. Death rates from smallpox, for example, in Boston, dramatically reduced. I do have to point out that Innocence Mather had begun some early variolation in Boston, even before the vaccine ever became available. But as you can see, that made an impact, as well as actually the smallpox vaccination, which was really an amazingly successful event. But again, how long did that vaccine immunity last? And Dr. Jenner actually had an opinion about that, and he published that. And he said, amongst the cases that I refer to, one will be found of a person who had the cowpox 53 years before the effect of smallpox was tried upon him, as he was completely resisting it. The intervening period of time, I conceive, must necessarily satisfy any reasonable mind. And so he also felt, he had no data for this, but he was a very, very observing clinician, clearly, and gathered all the data that he could. And he had already come to the conclusion that immunity could be long-lasting. So what did the vaccine do? Of course, it wasn't known back there in the 1800s. What is the vaccine supposed to do? And that wasn't known for about 100 years. And that came about 100 years after Jenner. And the first reference to anything called an antibody came from Emil von Behring and Kitasato in 1890. So this is now, many, many years later. And in their publication, they showed that the transfer of serum from animals immunized against diphtheria to sick animals could actually cure them. And the potential for treatment of humans was then immediately apparent. And Behring then later got the Nobel Prize for his work in 1901. And so it became clear that, in fact, you could transfer, but it wasn't known how that could occur. But of course, something in serum was creating this wonderful effect. So again, another very old print, which I think is very amusing as well, which was taking the serum off of this horse, because of course at the time it was a horse serum. And there's Dr. Behring in his laboratory, actually in his pharmacy, and he's giving that to, in this case, they look to be very rather happy housewives, and also this small child who's just coming up to fill her pitcher there with this lovely horse serum, which will give her immunization, in this case to diphtheria. But where did the antibody come from? Kind of moving along. Well, that was debated. Now here we are 45 years later, and Dr. Ehrlich and Harris, University of Pennsylvania, said in Science It's true that the lymphocyte is a somewhat prosaic cell with not particularly striking morphologic characteristics. If stained with routine stains, the cytoplasm seems to be singularly undifferentiated and unspecialized, especially when compared with that other white blood cells of the blood. In other words, not very exciting. And the lymphocyte probably is not involved in this astonishing process of making immunoglobulin, or gamma globulin as it was known at the time. Not clear exactly what cell was making it. And in fact, the phagocyte was the cell that was considered the most likely cell to produce antibody. Now, this is 1945, remember, so it's kind of interesting that even then it wasn't terribly clear where antibodies came from. So actually, the real view was that the phagocytosis of cells eating the bacteria, it would be most likely that the phagocyte would be the one making the antibody, kind of, one-stop shopping. The phagocyte eats the bacteria, and there you go. He makes the antibody afterward. Kind of an interesting conclusion. And that, of course, was, another interesting point of history, which is then, okay, well, where do they come from? And it wasn't really known until a couple of years later. And this is in her PhD thesis. Astrid Fagrius, who was doing her PhD at the Karolinska Institute in Sweden, had noticed that the horse serum injected into rabbits, there was an extremely close relationship between the plasma cell numbers in the bone marrow and the antibody in the blood. Interestingly enough, she also found out in multiple myeloma, there was an increase in plasma cells along with the hyperglobulinemia. And she herself, of course, was quite an amazing woman, clearly. Her PhD thesis then became immediately the dogma, which is that these lovely plasma cells are actually the antibody secreting cells. And so this, in a way, is kind of the opening salvo towards the world of primary immune deficiency. But the original 8-year-old child also clarified many other things as well, which is this is an 8-year-old boy who got admitted to Walter Reed Medical Center. And it's impossible to think, 19 episodes of pneumococcal sepsis, recurrent pneumonia, and as it turned out on the protein electrophoresis, he had also no immunoglobulin in the blood. And this was, by the way, one of the first times that serum protein electrophoresis had ever been done on a patient in the United States, because the equipment was not even available, except it was at Walter Reed because of the work of Arne Tiselius, who had come from the Karolinska to be at Walter Reed for a short period of time. And at the time, of course, we had no idea that the loss of antibody making was due to the loss of these antibody producing B cells. And that, of course, was due to the lack of this tyrosine kinase, which we've now named for Dr. Bruton, the Bruton's tyrosine kinase, BTK. Being on the X chromosome, of course, that's the reason why it was only males. And so this was really kind of an interesting wrinkle, because then it clarified exactly what was going on. And in several years later, then it became clear that in agamma-globulinemia, the same boys, that they had no plasma cells in their bone marrow either. So everything fit together. Now it turns out there's no antibody, there's no plasma cells. Yes, Astrid Fagrius was right. This is exactly what is going on here. I always look at this slide with great interest because you can see it was in the New England Journal of Medicine, so that means it was considered to be very unusual. But up until this very moment when Dr. Jay Sanford published this was a 44-year-old woman who got admitted to Massachusetts General Hospital. And here she's the wrong sex because it was considered that this was for young infants, male infants should be the ones that are agamma-globulinic because that was the way that the history ran. But here she is, she's 44, wrong age and wrong sex. And all of a sudden she turns out to have no gamma globulin either. Now looking back, we would end up saying, this is actually the first case report of common variable immune deficiency. And again, it turned everyone's notions again on their head because it's just the wrong patient. And how did she get along all this life? How did that happen? Of course, you can see she wasn't well during a large amount of this, period of time. But on the other hand, she was alive and did appear at Mass General, and she got admitted a number of times. So it does turn out that people can be agamma globulin and have no plasma cells. But again, where did the antibody come from? And where did the plasma cells, where did they arise? Remember that lymphocytes were not considered to be the source of plasma cells. And so this was kind of one of the other quirks of history. In 1956, the bursa fabricius had been known to be an organ present near the rectum of the chicken, but it wasn't known what the bursa did because bursectomized chickens were perfectly healthy. There was nothing wrong with them. But in the 1950s at the Ohio Agricultural Station, there was a graduate student named Bruce Blick who was studying vaccination of chickens, really because salmonella, of course, is a big problem in the poultry industry as well. And as it turned out that since there were no animals around the day that he needed them, he took some bursectomized chickens by accident and immunized them. But in fact, it turned out they made absolutely no antibody. And so he tried to publish that. He sent it to Science and said, you don't make antibody if you don't have a bursa. And it went to this, very high impact journal, but they said no. Further elucidation of the mechanism should be attempted for publication. So actually, the first publication about B cells was in Poultry Science, which I always think is a funny event. So it's really kind of an accident. I mean, I think I grew up thinking that B cells were for bone marrow, but actually it's not. It's actually for bursa from the chicken. Very, very odd, kind of historical quirk. And of course, it wasn't for another, 9 years. This is again 9 years later that it became clear that in fact there were two lineages of lymphocytes, the thymic cells, the thymus cells, the T cells as we would now call them, and then these bursal lymphoid cells in the chicken are actually translatable into human beings. And it turns out that there really is kind of a division of labor. What the thymus can do is one thing, and what antibody making B cells can do is a little bit of another. And this seems relatively recent to me to have separated these, positions in immunology into these two segments. So what does a B cell do? Kind of moving along and thinking about what the B cell is up to. A B cell is going to turn into this factory. And when I see these little organelles inside a plasma cell, it shows you all of the protein apparatus, which is ongoing. You have to remember that these plasma cells are producing about 20% of serum protein, which of course is a pile of protein all the time. 10 to the 8th B cells at any given moment. The naive B cells really don't live very long, between 3 days to 8 weeks. That's about it. If they have never met their antigen to continue to expand their lifespan, that's it. They die. But the plasma cells, which are kind of the factory here, they secrete somewhere around 1,000 IgG molecules in a second. Can you actually imagine that? That's actually been calculated from their quantity, from numbers of cells, and then measuring supernatants. It's kind of an amazing piece of work that was done to figure that out. About 2 nanograms per cell per day. So they're incredibly high energy, needy, first of all, and also very, very, very productive. So these are plasma cells. And this is exactly what's happening when we're immunized, is that plasma cells now are beginning to be formed. So when I look at this picture, it always strikes me as it looks sort of like the subway line, the Lexington Avenue line, and you have stem cells that just go to various stops. And, first thing you know, you're a plasma cell. It's kind of like very easy. The bottom line is they end up making gamma globulin molecules, which we usually draw as this Y-shaped molecule. It doesn't look like a hard job, right? In the bone marrow, they grow up and then they get produced out into the periphery. It's pretty simple. Looks like it should be just, one of those easy things. But think about it. You're going to build a gamma globulin molecule. You're going to build it from 150 immune globulin genes, which lie on 3 different chromosomes. So think about the assembly. It's on chromosome 14, 2, and 22. All these bits and pieces from the heavy chains and the light chains have to be reassembled, combined, checked out, again recombined, see which one works. Some work, some don't work. And you end up with somewhere around, 2,000 billion possibilities. It's a little hard for me to even think what that number would be. you end up with so many various combinations. And by the way, most of them are duds. Most of them will be discarded because they either didn't work, didn't join, there was a mistake, or they were autoreactive and they just got discarded. And this is ongoing, of course, all the time. And especially when you've been immunized, all of this has to take place. Now, this, of course, was the Nobel Prize winning of Jerry Edelman and Rodney Porter. And the two together then were, I think this was 1972, is the Nobel Prize for that work. Having said that, it's an incredibly error-prone process. All of these things which go wrong all the way through are going to then create a great deal of waste. Of course, the B cell then tries again and said, okay, fine, that didn't work. I'll do it again. I'll see what I can do about that. So this just takes some of this information and puts it into what's happening in the bone marrow. You're doing all this rearranging. You're taking the individual fragments from the heavy and the light chain. You're putting it together. If you can't make a kappa light chain, then the immune system says, fine, I'll go back. I'll do a lambda instead. Lambda is the fallback. You know, it's not the preferred arrangement or the preferred light chain in human beings. And it's just all of that will take place before the B cell ever gets out into the periphery. And then when it does, the next thing that has to happen is then it has to be continuously reconfigured, cut and paste and edited and realtered because it's not a very good fit for the antigen. The immunogen has already been perceived, but the fit wasn't very good. And so that was where somatic hypermutation takes place. And I think everyone realizes that the first antibody that you make is going to be an IgM. So the next thing that has to happen is it has to switch. It has to switch isotypes, again, be recarved up, and then reallocated, and then pasted back together again so that you can make your IgG and your IgA. And you could imagine that although an adult produces about 1 billion new B cells every day, it's just extremely error prone. So the risk of producing cells that fail to assemble is just huge. So they either don't assemble or they turn out to be autoimmune. And if they're autoimmune, they're going to be discarded as well, hopefully. If they're not discarded, of course, then we're going to be having an autoimmune situation. Of course, autoimmunity does creep through. And then this issue about somatic hypermutation, it's another very, very error-prone process, and it results in all these unwanted or dysfunctional B cells, which then end up in the trash can. So again, it's very, very wasteful. On the other hand, we know it works. It actually works remarkably well. The other thing to point out, and I like this slide particularly because this was 1996, and these are bone marrow samples that were taken by this author, Nunez, but he's actually working with Max Cooper. And if you remember, Max Cooper was the guy who first decided in 1956 the differences between T cells and B cells. And here he is in 1996, 30 years later, he's publishing on what bone marrow is doing in a person who is up to even 80 years old. And after the age of, say, 10 or 12, adults are continuously producing new B cells in their bone marrow. And all of these, of course, are bone marrow samples. So I'm not exactly sure how they got all these normal, healthy bone marrow samples, but they really do tell you that everyone is continuously making B cells their entire life. Of course, there's a burst in infancy, but then that burst then calms down, and it's very, very steady after this period of time. So again, we're making B cells the whole time. It doesn't change, no matter your age. You could be 90 years old and say the same thing. Now, this is one of those slides that you'll see it in a million textbooks, and it's actually on Google, of course. And so I just wanted to say a couple of words about, well, what is happening in that lymph node? How is that working? Well, everyone had the mRNA vaccination, and then that's going to instruct the immune system to produce the spike protein. So the spike protein is then going to be picked up by dendritic cells in the lymphocyte, and it's going to be presented to T cells. So T cells understand that this is really not normal. You have to do something about it. The T cells then instruct B cells the same way, and B cells then have to take a journey. They have to go from the exterior of a lymph node into the interior of a lymph node, where they again meet T cells, and they're given a chance to bathe in a number of cytokines. And in this process, they're attending really what's an intensive training period. And this particular notation up here just says all the intimate contacts that have to take place between this germinal center B cell and all the other cells, T cells in this case, that takes place inside a lymph node. And what happens? At that time, then these germinal center B cells then are trained. They understand that was the immunogen. This is my life journey. This is my life. This is what I do. And I'm a memory B cell. I will memorize that spike protein. It's my job. No matter how long I live, it's my job. And some of those cells then turn into plasma cells, similarly instructed that that's a lifetime's opportunity. This is kind of the big picture of the same thing, and I'll show you again in a moment the timing. In about week 1, this takes place in your first week, these very short-lived plasma cells will begin to secrete some IgM very quickly. So after your first spike injection, you're going to be having IgM in about 1 week. You'll be having some protection even that early. In week 2, the cells are migrating into the lymph nodes. That's why your lymph nodes felt the way they did. That's why perhaps under your arm it felt sore and tender, because there's a lot of activity going on at that time. And then, of course, we have memory B cells and then the plasma cells. And we have what we have now learned to call long-lived plasma cells. And that, of course, what I was talking about when it comes down to that smallpox memory a moment ago. Now, when you look at a slide like this, I know it looks a little fanciful, but if you actually can look at it visually, this is a real lymph node here studied by immunofluorescence. And it's pretty striking that you can actually label that lymph node and see what is going on in the lymph node. So these are going to be young B cells. They are B cells that are staining with the young IgD molecule, meaning they haven't really learned their lesson yet. They haven't gone into the germinal center to learn what they need to do, but they're ready to do that. These are very early plasma cells. This is an area called the follicular zone. This is an area called the marginal zone of B cells. And this, of course, the hotbed of information taking place here. It's pretty striking when you look at the cartoons of cells and then realize you can actually look at it on staining, and you can prove that really is an operation that's taking place. Now, this is kind of a boring slide, and so don't take it as anything other than when you look at memory B cells, curiously enough, they all have markers. In other words, a naive B cell has a certain look, and it has certain markers by immunofluorescence. You can stain those cells in the blood and say, what family do you belong to? Are you naive? Are you naive resting? Are you naive activated? Or are you a cell that's just exhausted? Say, don't tell me anything else. And that's an anergic cell over here. These are going to be the memory family over here. And again, you can tell what stage they're in by all these markers. And you can do this in anyone's blood at any time. It's pretty amazing, really, that we have all these interesting phenotypic markers that will tell you exactly what the characteristics of each of these cells could be. And then, of course, the antibody secreting cells are quite different. And this CD27 here is a marker of a cell which has got its diploma. I always think about that marker as sort of the diploma. It graduated, and it means that I know my job. And that means, of course, it's never going back. It will never change its mind. It can only do that one thing. Again, just to say something about the fact that when you say that these cells are different, they also have mRNAs that are very different. And so not to mention what any of these are in particular, because that's not important. This is a naive cell, which has a lot of genes that are just basically turned off, or turned on, I should say. And these are cells which these are markers that say, I'm quiet, nothing is happening. Green, of course, means quiescent, and red means turned on. But the opposite was true for the memory B cell. In other words, it's really a completely different mRNA construction of these cells. They have lots of genes turned on or turned off during that process of developing, and you can mark each of these, and they all, of course, turn out to be important molecules for how things work. And you can boil that down into also cytokines and growth factors. And again, the IgG memory B cell has certain things which are just very, very positive, and other things that are just completely shut down. And it's just interesting that, while we look at B cells as growing up and having certain activities, you can actually also stage them. So how long is antibody memory? We keep talking about how antibodies and immunoglobulins are being enforced upon the immune system, and B cells are getting more and more informed, and they are getting better and better at their job. But how long does it last? And I think this is one of those amazing slides that will probably never go out of fashion. I don't just have used this slide before because I think it's pretty astonishing. This is Shane Crotty and Rafi Ahmed, and at the time, I think they were in Atlanta. And these are taking the serum samples of Army recruits who had been immunized to smallpox previously and looking in their blood. Again, this is a log scale. You can see that up to 60 years after being immunized with smallpox, there's still antibody. And it tells you that that vaccine memory can last perhaps almost a lifetime. Of course, individuals who were never immunized do not have that memory. And then going back, I talked to you about memory B cells. You can go back and you can ask the B cells in the blood of those subjects as well, do you have memory B cells? And the answer is yes. Those who have a large amount of IgG antibody have lots and lots of antibody cells that recognize the smallpox. Now, smallpox is not something you can just run into, inadvertently just by having someone sneeze on you or inadvertently meeting the antigen so those cells could be kept alive. Smallpox is actually not available. It's not something which is around. It's an antigen which is not, it's in the freezers, for example, in several locations in the world, but it's not readily available for increasing immunity. So this is lasting immunity. So these persistent smallpox titers really lasted 60 years after exposure. Pretty amazing. And so that's important. And what else do we know? Interestingly enough, there's also plasma cells in the bone marrow. And these are the producers of that antibody that you see in the blood. This is another study looking at some of the same individuals, saying that the survival of smallpox-specific plasma cells in bone marrow 40 years after vaccination, again, can be found. So again, the plasma cells are keeping that memory. The memory B cells are keeping that memory. And so that we actually, come down to some knowledge about exactly how this works. This is another interesting slide, too, making the same point, which is that what about the 1918 influenza epidemic? Again, the same story, which is that up to 30 years after having that, recalling individuals who had experienced that influenza, you can still find both antibody and, curiously enough, and interestingly enough, from our standpoint, of course, is neutralizing antibody. That would be the green bars here. So these are very, very functional antibodies. Again, very, very long-lived. And there's another one to show you, and I don't know whether these vaccinations really are, you can see them very well, but it doesn't matter because the point is that these are an amazing, I think it's 630 samples from 45 different people were gathered, and they were examined for antibody to vaccinia, measles, mumps, rubella, varicella, Epstein-Barr virus, tetanus, and diphtheria every year for, I think, 35 years. And you can see that all people had, for example, no measles, mumps, or rubella vaccination since they were 5 years old. Here we are, this person is 50 or 60 years old and still has antibody to all of these agents. These are remarkably consistent titers if you look at it. So again, it says, well, all right, accumulating your information, you had smallpox, you had flu, measles, mumps, rubella, chickenpox, tetanus, and diphtheria. These are very, very lasting immunological memory, which has been created in all of these individuals. Again, a wonderful article in the New England Journal of Medicine, which again gives us some feeling of how long things could last. And we know that's the plasma cell. Now, one thing to mention is plasma cells do not divide. And what that means is the plasma cells that you had when you were 5 years old are the same plasma cells that you have now. They're not going anywhere. And once instructed, they can last perhaps an entire lifetime. Memory B cells, similarly. We now believe that memory B cells can last as well also a lifetime. Now, if you look for the lifetime of other cells, and I gathered this off various websites and things, only neurons really beat plasma cells and B cells. Neurons, you keep your entire life as well. Heart muscle, maybe 40 years, that's wonderful. But what about other cells here? As you can see, these cells here actually amazingly long-lived and much longer than other cells of the body. And again, that's one of the reasons that I think, this particular study of how immune system works and vaccination is something which tells us something which is really pretty unique. Now, you could imagine that a lot of people have spent time thinking, gee, that's a long-lived cell. How does it manage that? How does it do that? And of course, that's because the bone marrow niche has a number of mechanisms for keeping plasma cells alive. This is a plasma cell. And part of it is the extremely close contact with various molecules, each of which is necessary for adhesion and holding that plasma cell in contact, and a lot of other adhesions over here, as well as cytokine CXCL12, for example. APRIL is produced by a number of lymphocytes. APRIL is a proliferation-inducing ligand. It's an interesting cytokine that causes plasma cells to live a long time, binding through several receptors. BCMA, for example, and the TACI receptor bind to this APRIL. This is something that we are interested in our laboratory here, IL-6, for example. And those are being produced by all of these cells. I didn't realize eosinophils are actually the strongest producers in the bone marrow of APRIL. Makes you wonder whether that's really the job of eosinophils in life, is not to cause allergy, but to keep your plasma cells going. It's a very interesting thought. So this is a very special niche that has to be developed. And the other interesting thing is that all the things that I said a moment ago, the plasma cell can actually secrete up to 10 molecules per hour, which is 2 nanograms per day. You could imagine that a plasma cell actually has tremendous energy requirements, right? With all of this production of protein, a lot of glucose, amino acids, and short-chain fatty acids, number of differentiation factors, all the other things down in here. And they also have to be kept rather hypoxic, which I find very interesting. They really have to have low oxygen. You give them a lot of oxygen, they die. Very interesting. Now, you could imagine quickly that one reason for thinking about plasma cells and knowing so much about them also has to do with myeloma. In other words, how would you deprive a myeloma plasma cell from their necessary factors? And of course, each of these has been the target of oncology as well, because you, in some cases, want to keep your plasma cells going, and in some cases, you have way too many of abnormal ones, and you would like to have them disappear. And so all of these things are something which of really great intense interest in oncology as well. Now, from my standpoint, what can go wrong and why? Again, this looks like a simple job, doesn't it? Looks like it's pretty easy. But there could be a situation of having no B cells, no B cell activation, no maturation, no plasma cells, and no antibody. And of course, there's many, many immune defects. The only one I'm going to talk about today is the primary immune defect. Of course, cancer will do the same. Chemotherapy, transplant, protein loss, malnutrition could also hamper any of these. But just today, let's talk about this. At Mount Sinai, we've been particularly interested in the primary immune deficiencies. And this happens to be Mount Sinai data, but if you go to any textbook on immunology, this is about the breakdown of every major center that studies these primary immune defects, which is that the antibody defects are actually the more common. So those would be hypogammaglobulinemia, IgA deficiency, IgG subclass deficiency, X-linked agammaglobulinemia, and moving down to neutropenia and a few other things. Some are rare and others are relatively common. But the interesting thing to me is that all of these marked in yellow have problems with antibody production. Again, it's not just the antibody defects, it's some of those which have some of those T cell defects as well. Because if your T cells are not working correctly, then your B cells never get their appropriate signals either. And the whole thing really goes together. So this group, in terms of what we see here at Mount Sinai, and in fact, every other major medical center, is the antibody defects actually are more numerous. And these just outline kind of the basic names of those ailments: the agammaglobulinemia group, the hyper-IgM, which means they can't switch, they never can make anything but IgM, and then common variable immune deficiency, which is really anything from this transitional B cell all over here to this plasma cell. Many, many IgG defects where their IgG is not correct. And of course, IgA deficiency, which we still have no idea what the actual gene cause of that is or gene causes. And then just antibody defects. In other words, somebody who doesn't make an antibody to pneumococcal vaccination, but everything else is pretty okay. And this has been an area of really quite a bit of investigation, all the things that cause your B cells to not work. And you can clump them together in hypogammaglobulinemia, or in this case, if you do a PubMed search, this is common variable immune deficiency. And ever since the first article came out over here, that's the 1954 article, slow increase in the number of publications, because this area turns out to be very fruitful for several different reasons. It tells you a lot about how normal human beings produce gamma globulin. At Mount Sinai, we, of course, see quite a number of patients with hypogammaglobulinemia. We have 741 or so CVID patients. As you can see, the IgG levels are quite low. Normal is greater than 600, of course, for IgG. So these are quite low numbers. Usually the IgA and the IgM are deficient as well. I think one of the stunning things to remember, and this is why I think it's a good thing to remember for any internist, is that these are not children for the most part. 20% are under the age of 21, but most people are not children. Most people are not diagnosed until they're older. Males are sometimes diagnosed a little bit earlier. Our average age is actually 25 for males, but females are really much, much older than that in most cases. So it goes to show you that this diagnosis category is one which is started off in the pediatric world, but remember, it's really largely an internal medicine job. Now, I said these are genetic, right? And we've been considering them genetic, and they've been included in the genetic listing now for the last 60 years or so. But okay, what are the genes? And these are some of the genes. And as you can see, I didn't even start the listing sooner than 1990, because that's really the first time any of these genes were actually cloned. And so here we are with the first one is BTK. And so that's Bruton's tyrosine kinase, discovered in 1952, but it was actually 40 years later that the gene was cloned. This is the gene for hyper-IgM syndrome here. And moving on over here, you can see that there's been really a cascade of additional genes. All of these are the ones that are important for making antibody. And of course, we're all using these genes every day, whether we know it or not. They are actively moving along. Now, as you probably have realized, I'm very interested in the topic of hypogammaglobulinemia. And this just gives you a quick slide of a very, very large amount of work, which was published in Blood a year or so ago. And that is that this is the Mount Sinai data over here, looking at the genetics in terms of our particular patient population. Out of 235 whole exome sequenced patients, 31 have relevant mutations. So I look at that and I think 31 is amazing, considering where we used to be, which is 0. But 31 makes me think, okay, fine, that's wonderful. What about the other 70%? Where's all the other data? Tell me what the other mutations are that create this particular phenotype of hypogammaglobulinemia. Now, my colleague Lennart Hammerstrom at the Karolinska also did something similar with 128 of his patients. He then sequenced those patients as well, found 36 with relevant mutations. And again, the breakdown of the types of mutations is somewhat different. He has a few more autosomal recessive genes than we do. And then my Iranian colleague, Hassan Al-Busani in Tehran, did 235 patients. And again, all of these patients would come in the door and be diagnosed with common variable immune deficiency. But you can see the genetics are different. They're not the same with each. And to sum it up, there's 56 genes in 601 subjects, all of whom who are hypogammaglobulinemic. In the United States, the autosomal dominant genes with variable penetrance are the most common. You often get a family history, but you often do not. Autosomal recessive are less common, and a very few X-linked will creep in. A little bit different breakdown in the Swedish group, and then much more recessive genes in the Iranian group. But it tells you that all of these genes are how we make antibody continuously. It's how we do it. And here I've just taken that original slide and put some of the gene names on here. Something can go wrong anywhere along any of these, I would say, subway stops, so to speak, starting with the genes that don't allow you to even make a B cell, and those genes which allow you to make a few B cells that don't work, and then some that allow you to make some B cells that look pretty good but don't make antibody. And moving on over to this one here, which means it's making a B cell, it's just the B cell can't crawl out of the bone marrow. So it's just another one of the very many ways in which immune system is producing this amazing factory, I would say, is with the use of all these genes. And this slide just, again, says the same thing. Tremendous number of small interactions have to take place, each of them correct. And so all of the genes that are marked on these slides are then targets of our interest, partly because all of them will give you a result, which means no antibody can be made one way or the other. All these interactions are necessary. So conclusions. Well, how long does antibody persist? So what do we know? Let's gather up what we do know. The half-life of serum antibody against protein vaccines, and now we're talking about our tetanus and diphtheria, is going to be 10 to 20 years. That's the half-life. It doesn't mean it's gone. It means it's half gone in that length of time. And we know that from that study of Amana et al. In the New England Journal of Medicine. Now, what about antibody to live viruses? Measles, mumps, rubella, vaccinia, which is smallpox, chickenpox, Epstein-Barr virus. How long is that? These are very long. Between 50 to 200 years is the half-life. That's the calculated level. Now, what about rapidly mutating viruses? What about something like Ebola? That's not clear. I don't think I know exactly how rapidly mutating Ebola is, but we do know the COVID virus is actually mutating. We are already aware of that. So how much will the immunity that we've been given last? That is less clear. We have to be clear about that. And what about RNA vaccines, mRNA? We have limited data, but there is suggestions of long-lasting B-cell memory. And the reason why is that this is a picture from another review from Andreas Radbruch in Berlin, who has also studied bone marrow. And as long as we are currently able to do so, realizing the COVID pandemic is really only about 2 years now, as far as we are currently aware, it does appear that there are long-lasting plasma cells that will confer long-lasting immunity, even in our COVID world. So taking what we know and taking what we surmise, we suspect that what we'll be getting is really quite a bit of continuous immunity, but it will, of course, depend upon the mutational rates. I just put this slide in because, to me, it's kind of this historical overview of smallpox vaccination down to the first time a BTK gene was cloned. And all of these are sort of, I would say, guideposts along the way to understanding a little bit about how we make immunity and how we keep immunity and how we keep our B-cell immunity and how it works and why does it go wrong. And of course, as you might expect, these are all Nobel Prizes that were given for various reasons along the line. Each of these would be areas of Nobel Prize winning work. We depend very much, of course, the work of Cesar Milstein with the first hybridomas made. And of course, that, of course, is how it is that we have some of the monoclonal antibodies. And that, of course, was Nobel Prize in 1975. So these are some of the very wonderful people that I have worked with at Mount Sinai, including my colleagues at Rockefeller, which is Jean-Laurent Casanova and Bertrand Bosson, as well as in my laboratory currently is Jose Chavez and also Yolanda Carmona Garcia and also Lynn Radigan. We're hoping very much that PJ Maglione will rejoin us. I have to mention some of my previous MD PhD students, which are here this top row, and that would be Patrick Mafucci, Noah Simchani, and Tom Marion. And these are really wonderful colleagues, and it's been absolutely a pleasure to work with everyone at Mount Sinai. And just to put down a few other names, these are some of my other colleagues elsewhere. So thank you very much for the opportunity to give this Ludwig lecture today. I appreciate being asked. Thank you very much.
Thank you, Dr. Cunningham-Rundles. Wonderful talk. Let's see if we get some questions in the chat. Dr. Greenberg, you always have a question, Dr. Greenberg. We have a lot of participants today, Charlotte, so people were very interested in your topic.
I think everybody wants to know about COVID.
Yeah, right, exactly. So we do have can you comment on the T-cell immunity as a question?
I wish we could. There have been a bunch of articles about that, and we've been actually studying it in the lab. So if anybody wants to help us, being a normal volunteer who got immunized, we could use more normals. At the moment, we've been using our fellows, so and also ourselves. But you do find in the blood about 1% of T cells that are, if you expose them to spike protein, you discover that they can make interferon and interleukin-2. We're not completely ready to go to the mat with that, but it looks as if that's pretty much what others are finding too. How long does it last? I'm not sure, but they certainly do recognize they do recognize spike protein for sure.
There's a question. Do you think we will need annual vaccinations like flu vaccines be recommended for COVID?
You know, I wish we really knew. I suspect that we will do that until we're absolutely confident. My bet is that we currently do have a great number of plasma cells already started and already launched. I think especially for people who've gotten those 3 vaccinations, we probably do have now instilled a lot of vaccine memory, I think. You can detect so much antibody in the blood. We've been doing quite a bit of antibody seeking, and if anybody wants to know the best test, I think we know which one is better than others for checking antibody titers. I think as long as we can still detect it in the blood, that certainly means we've got plasma cell memory. I think what we don't know quite is how much mutations will take place. We already know of the mutations from the Delta to the Omicron, but not being my area, it looks as if, you know, one version is just somewhat weaker. How many more mutations could occur, I think, you know, is really open to question. We can't project.
Gotcha. Which cell population is different in patients with prior COVID-19 or other versus those who got only a vaccine?
It seems as if there's higher titers in those who have been doubly immunized. Yes, that's true. Whether or not they will end up being a big difference, I'm not sure. The only data that we have seems to show that after 3 vaccinations, the titers are approximately in the same territory. And so that's not clear. I don't think we are aware of that. Whether the vaccine memory will last longer when you've also had COVID is possible, but there's no data on that.
Right. We have some questions from residents. So what advantage does switching from IgM to IgG confer?
That's a really interesting thing. IgM is a more expensive molecule if you think about it, and it's shorter lived is one of its problems. IgM really only lasts around 5 days. IgG lasts more like 25 to 30, more like 30 actually. So it's a bit of a longer lived molecule. And the other thing is that although IgM is a wonderful molecule, it's not able to get into all tissues either because it's rather larger. So you could make it up, but IgG seems to be the more workhorse molecule, honestly. You could ask also about IgA actually too, couldn't we? Like how much IgA antibody is there? And I think Florian Kramer has measured IgA. It could be it's also very useful for mucosal surfaces. I've not seen any publications on that particular part.
Gotcha.
It may be out there.
Questions from some residents. Do we know how CVID results in nodular regenerative hyperplasia of the liver?
We have a hypothesis about that. So if anybody's interested in what we think, we published in I think it was November of this year that the patients with common variable immune deficiency have a great deal of bacterial DNA in their blood. And it's 16S DNA, which is universally present in all bacteria. So we then speciated it, so we know where it came from. It came from the GI tract. Our view is, and my hypothesis is that's why we have nodular regenerative hyperplasia in the CVID patient population, because there's an insult to bacterial DNA. And we are quite convinced that there's a marked increase of gamma interferon signature in those bloods as well, in the blood of those patients. So you can see that there's a cytokine insult almost. We're very interested in pursuing that actually, although it's going to take a bit more time.
Gotcha.
Yeah.
Another question. If smallpox immunity lasts so long, why did the government want to vaccinate healthcare workers and first responders who had a primary immunization as children after 9/11?
It's probably just the usual story, abundance of caution. Perhaps maybe they didn't, maybe it wasn't quite as good. But if you look at the data said that it was extremely long lasting. The data, I don't know if the government wanted to go out and measure titers on everybody, probably didn't, just thinking, hey, just give them the vaccination and call it a day.
Oh, gotcha.
How would you, it's like when you go to college, you're asked, have you had all these vaccinations? They never take a titer. They just ask, did you have the vaccine? But they don't really look at the data. Right?
Gotcha. So there's a comment and a question. Thank you for this wonderful lecture, which I agree. How strong is the data for plasma cells really living for such a long time without proliferating? Is there any evidence for homeostatic proliferation to keep them going or replaced from memory pool?
Being replaced from memory pool, you can't exclude it. But the data on plasma cell and lifespan comes from BUDR data incorporation. And you can do some studies, and it's been done about that. I think one of them was based on radiation molecules, radiation studies from rheumatoid arthritis patients. And when you know the date of the radiation event, you can go back and then backdate the plasma cell. There's some very interesting data on that, which I would hesitate to quote anymore because I don't want to give chapter and verse on that. I think the data on it's not reproducing is extremely strong. I think that's kind of an accepted fact.
Gotcha.
Is it possible that some of them are creeping over from memory B? Can't exclude that.
Another comment and question, Charlotte. Super talk from Dr. Kohler. An Omicron-specific vaccine is being tested in Israel now. Mm-hmm. If it produces good neutralizing antibody and is safe, would you recommend it?
I guess we have to say yes, don't we? Do we have a choice about that?
I guess so. I think that's the last question. I think. Yeah, that's the last question. So, a great talk, Charlotte. So thank you very much for coming today and talking to us. Congratulations on receiving the Ludwig Award today. So have a great day, everybody. Thank you all for coming.
Thank you very much.
Enjoy your day. See you, Charlotte. Have a good day.
Thank you.